So our speaker today, I'm really excited to introduce Jason Manzon. Jason is a 4+1 master's student in mine and Dr. Stephanie Arwood's labs. And so the backstory here is that in 2023, in the spring of 2023, Dr. Yarwood and I received a grant from the Hughes Center for Agroecology to look into climate impacts and how shifts in soil moisture could affect carbon stocks, especially when it comes to using cover crops, winter cover crops. And so I'm going to let Jason talk about that. That's kind of the backstory. We opened it up as a capstone project initially to see if we could get some undergraduate interest and just get some people working on certain aspects of the project. And Jason was one of the undergrads who responded to it and showed a lot of interest, so much so that we got him on board for a 4+1 program in the department. And I'm really happy we did that because he's done a great job. And he came with a lot of really nice qualifications to begin with as an undergrad. He did some work with the Joint Institute between the university and the Pacific Northwest National Lab. He also did some other internship work with the Department of Energy and also had the Holland Scholarship from NOAA. And even had a publication, I think, from some of that work involving soil respiration rates, like on a global level. So really cool stuff. And he brought that work ethic and that experience base to the work that he's going to present today. And a few things I'd like to say is that as a mentor, Jason has done a lot, I think, to mentor students in our department, undergrads especially. There will be undergraduates in the future that will become graduate students because in some way, shape, or form, Jason has helped mentor them and put them on that path. So I want to thank him for that. And I also wanted to say that the work that he's going to talk about today, he's also shared important components of this at international conferences like the Goldschmidt Geochemical Conference, the American Geophysical Union Conference, and most recently at the Soil Science Society of America. And we've got way more interesting data to talk to you all about today. So with that, I'm going to let Jason take it.
All right, so today I will be discussing a little bit about the second chapter of my thesis and a little bit about the first. And so the second chapter is titled Mechanisms of Soil Carbon Sequestration in Winter Cover Crops Driven by Differences in Soil Moisture. Jared mentioned my name is Jason Manzon. My committee is Dr. Stephanie Arwood and Dr. Matt Fischel from USDA and Jared. I'm actually co-advised by Stephanie and Jared. I'll start with the background to get you all caught up on what we're going to be talking about today. I'll go through a little bit of chapter 1, which is the soil fractionation experiment, just to introduce some methods that will become relevant later. And then chapter 2, cell processing and the results from it. All right. So in general, wetter and warmer winters are projected to occur in Maryland. The plot shown here is a NOAA output showing relative to a 30-year baseline, medium emissions and high emissions scenarios for precipitation in Maryland in winter months, December, January, February. When we're thinking about cover crops, we don't really think of the winter as just being December, January, February. Usually it's something like October to April, October to May. But these trends still persist into the fall and the spring. This trend is visible in historical data as well. This plot shows relative to a 60-year baseline changes in precipitation. So what could wetter winters mean for agriculture? In general, as soil moisture increases, we understand soils to cause cover crop physiology changes. And that could just be the height of the plant, the amount of above ground and below ground biomass. Changes the cover crop kill date, which would be a land management decision made by growers based on, sometimes especially in areas that are not as wet as we are currently, based on the soil moisture. Changes to the rate and carbon additions to the soil, and that can happen primarily via phylo deposition, so it's leaf-litter drop, above ground biomass, or rise of deposition, and differences in how photosynthate C, photosynthate carbon, is partitioned in the plant soil. I'm going to be referring to carbon as C in this talk, so C in carbon. And what are photosynthates? It's any CO2 that's taken in the plant via photosynthesis. So photosynthesis, photosynthetes. It's carbon assimilated from the atmosphere. And how these are partitioned in the plant and soil has been a discussion of research for the past 25 years as technology's gotten better for analyzing these things. And that's what we're going to be talking about today. But in general, there's a lot of uncertainty surrounding best land management practices in the face of climate change, right? So you're trying to manage all of these factors. What do you do? Let's get a little more familiar with agricultural systems and how carbon cycles within the agricultural systems. This is relevant because it's separate than a lot of less managed systems like forests or permanent grasslands, where you have constant roots in the soil. Some agricultural systems you might have constant roots as well, but there's nuances. So inputs, you might have deposition, right? The phyllo and rhizo deposition that I talked about before, organic amendments like manure. Microbes and fauna also add a lot of carbon to soil. And when the carbon gets into that soil, we generally think of it as being within two broad classes of carbon. This goes into how we characterize carbon in soils. We do it based on what we see, what we measure. And usually you want to characterize soil carbon in a way that lets you recommend something about land management, right? And so, broadly, people give these things many different names. We have weakly held or free and easily accessible pools of carbon, and our stable or recalcitrant pools of carbon. When I say accessible or stable, I'm talking about relative to microbial decomposition. Microbes will chew up organic matter and release carbon as CO2. That decreases the amount of carbon in the soil. It's not a one-way street, but these pools can interchange with each other. A little bit of an overlap in my diagram. But microbes can decompose organic matter, and over time it can become smaller, where it's easily more stabilized on the mineral surface, or put inside of a micropore where maybe it's excluded from microbial activity. And so these stable or recalcitrant pools, especially in agricultural systems, can be released into the weakly held, free, and accessible pools, accessible to microbes. And a lot of that happens because of tillage. Tillage can disturb aggregate structure, cause slaking of aggregates, which releases this stable organic matter into the free pool, perhaps. Respiration and tillage can cause a lot of CO2 release. Removal of cover crops from agricultural systems, any crop from agricultural systems also removes a significant amount of carbon. You also have runoff, which removes carbon from any system. And so when you're trying to, if you're a grower trying to balance the amount of carbon going in and out of your system, you probably want to choose land management practices that keep things cycling instead of having mostly exports of carbon from your system. So there's great potential for carbon to be stored in agro-ecosystems. We think about a forest relative to agriculture. Forest, right, I mentioned permanent roots. You might have a lot more carbon in the soil. If you think about, the way I like to think about it is that, let's say you have just a cube of soil in front of you. That cube of soil can only hold so much carbon, right? Before the mineral surfaces and the aggregate structures, they don't allow for any more carbon to be held. In agricultural systems, this maximum limit of carbon is not met, right? Because we're tilling the soil, we're growing things in the soil. I'm not saying tillage is necessarily a bad thing, there's benefits to tillage as well. What I am saying is there's potential for carbon to be stored in agricultural systems. So I'm going to characterize organic matter in a way that's been more common in recent years. And I'm introducing this here because I thought it would be good to introduce the background of what these pools mean. And that's what I'm going to be talking about the rest of my talk here. So people have latched on to this idea that there's two prevailing pools of organic matter in soil. The particulate organic matter, or POM, and there's this larger, more transient and easily accessible energy for microbes. And it falls in line with our weakly held or free and easily accessible pools of carbon. This diagram from Mutual 2023 explains it pretty well. It's defined by size. Any POM is greater than 53 micrometers in size. And there's mineral-associated organic matter, which is bound to minerals. It can do this partially because it's smaller. It's more easily bound to minerals. Has potentially decades to centuries of residence time, and typically takes longer to form than POM. It also acts as a reserve source of carbon and nitrogen. When things like tillage disturb agricultural soils, they disturb the aggregates within that soil. So you have your mount, less than 50 micrometers in size. Reliably characterizing these soil organic matter fractions can help us determine the effects of land management and climate change on agro-ecosystems. So now I'm going to go into a little bit of the idea behind chapter 1 of my thesis, which is a soil fractionation experiment. We just mentioned characterizing organic matter, right? There's downstream effects, though, to how you split out this organic matter. If you have a soil and you're trying to split out this BAUM and this POM, depending on what kind of extraction you use, you're going to get different results for what is in the soil. So if you're trying to, and this is specific to if you're trying to characterize the carbon in the soil, right? Let's say you're trying to characterize what people call a native carbon, or the native organic matter in the soil. That's before alteration. Traditional chemical extractions can alter the molecular carbon chemistry. This isn't necessarily a bad thing. It's fine for measuring total carbon, but if you're looking at molecular analysis, then you could run into some you make some assumptions, there's some caveats. Physical fractionation methods could help us better characterize soil organic matter though. And so what we decided to do is to look at this method of sonication, or ultrasonication, for splitting organic matter in soils into mound and pond, these two identifiable fractions defined by size that have implications for land management. So what is sonication? You can see it in the middle panel here. This probe dips into the soil, it vibrates, releases ultrasonic waves, and these ultrasonic waves burst aggregates. There's cool mechanisms for how this happens. I'm not going to go into it, but ask me at the end if you're interested. So that's the idea. So this is our physical fractionation method, and the undergrads who work in the lab are intimately familiar with this. I've done probably a couple hundred of these. So we have our soil. I'm going to go through really quick. We have, usually we start with 10 grams, sometimes 5 grams of soil. We skim off what's floating on top. That's the free palm. It's characterized by being less dense than water. And then we apply the low energy sonication step. This low energy sonication step is meant to disperse larger aggregates in soil, because larger aggregates are not as mechanically stable as smaller aggregates. So it takes a high energy sonication to disperse these smaller aggregates and lower energy to disperse these larger aggregates. There's nuances to why we do it in this order that I don't think is relevant today. But we use a low energy sonication to release all of the palm from aggregates. We use a high energy sonication to release all of the mound from aggregates. We actually split those micro aggregates that hold the mound into core silt and mound. We do this because we know them to be chemically distinct in some ways. And so I mentioned just palm and mound before. Now we have these 4 different classes. And again, here's the idea. Using sonication to split into different fractions. I put here optimal energy in H2O. What is optimal energy? Well, depending on your soil's mineralogy, the organic matter percent, a bunch of other factors, you have different ideal energies that the sonicator can deliver. So if you use an energy that is too high, you risk breaking up organic compounds. You risk breaking up larger organic compounds and putting them into a size class that they're not supposed to be. Right? Let's say you have something greater than 52 microns in size. That's the O-palm fraction. And you use sonication at very high energy, and then you destroy all this organic matter, and then it ends up in the mound fraction. Well, it's not what you want. So there is this target that we were looking for. So what we did first is we decided to optimize the low energy sonication. That's shown in this plot all the way on the one side. It's kind of condensed in there because this is a large x-axis scale. Let's talk about this x-axis scale. It's in joules per mil. That's a function, that's a units of energy for the sonicator that's delivering. And we did a gradient of sonicator energies from 0 to 500 to test the optimal energy to disperse our soil in particular. As part of this project, we dispersed another soil as well. We're going to compare them, but it's not relevant right now. The y-axis is the mass recovered from these samples. So for this low energy sonication, we were trying to look for increases in the OPOM. We were trying to look for any change in the OPOM, really, to see what is the optimal energy to disperse it. That's the blue line up there. There's no significant difference between those 4 points. What we decided was that our soil is pretty weakly aggregated, right? In agricultural soils, especially around here, where you have one-on-one clays, typically you can get pretty weakly aggregated soils. So we think that dispersing the soil and water got rid of most of the water unstable aggregates. But we decided that the first sonication step would be 15 joules per mil because that would be enough to get any water stable aggregates that might still be in there. Then we sieve the OPOM out as part of the procedure. And then we sonicate those micro aggregates, those smaller aggregates. We split those up. We split the mound from the co-silt. These are all held in micro aggregates. And this is a flat line. This plot is basically showing us that the optimum energy is somewhere in between that end point for the first section, which is 60 joules per mil, and this first point for the second section, which is 150 joules per mil. So we decided 150 joules per mil to be the optimum energy to disperse micro aggregates in the soil. And what is optimal? What are we looking for here? We're looking for bigger stratification between the co-silt and the mound. If normally these are held together in micro aggregates that are larger than mound might be, so the co-silt line decreases because you're splitting up the aggregates that hold both co-silt and mound. I think that makes sense. Let's get into this is the meat and potatoes of the project. This is chapter 2, the greenhouse 13C labeling experiment. So if you remember from before, right, we have palm, mound. Our goal is to investigate the mechanisms of soil organic matter formation under different moisture conditions. Moisture conditions because of this idea that Maryland winters are going to get warmer and wetter. We want to be able to link soil moisture and cover crop type to carbon storage and stability. We want to figure out where within the soil organic matter is carbon being stored. This is relevant for land management practices as well. So the mound is known to be, again, that reserve source of carbon, the reserve source of nitrogen. It's stable carbon. And in an agro-ecosystem where you have potential to build up carbon, you want to be putting carbon into your soil in a stable way. Right? You don't want it to be totally transient. You want to build the mound stocks a little bit, just in case maybe something happens. Right? And so we're trying to answer, where is the organic carbon being stored? How does the stability of carbon compare between soil organic matter fractions, moisture regimes, and cover crop types? This question of the stability of carbon is more unique to our experiment in particular. And we're trying to figure out knowledge gaps surrounding organic matter formation on short time scales. Relatively, generally it's understood that mound takes a long time to form. Well, more recent papers have shown that root exudates, for example, being very small, are easily sorbed to mineral surfaces and held there tightly. And so with better technology, maybe you can measure mound at a finer scale. We're trying to find out that mound can be formed in a relatively short time scale. So our hypothesis is that elevated moisture will enhance carbon sequestration to a point as a result of increases in biomass, and that extremely wet conditions will hinder carbon input to the soil as a result of hindered biomass accumulation and carbon deposition. And that's shown in our experimental or illustrated hypothesis here. From top to bottom, we have different water-filled pore space, or WFPS, referred to as water-filled pore space treatments here. So we're applying different moisture treatments to these soils in these undisturbed cores of soil. An undisturbed core is, if you do a greenhouse experiment or a pot experiment, typically you're going to have disturbed soil, soil that you homogenize, put into a pot. You're trying to observe some mechanism. But when you have undisturbed cores of soil, you preserve the pore structure, mostly you preserve the aggregate structure. And so it's a little more, you make a little more assumptions about how the soil might act. So we're applying different water-filled pore space treatments, 35%, 45%, and 65%. What is water-filled pore space? Of all the pores in the soil, it's how much of that pore space is filled with water. We chose 35% as the normal, and I hesitate to use the word normal here, but this is what I found from scan data from NRCS to be somewhat normal for the 20 centimeter zone in our region. Elevated moisture is around 45% because this is around microbial optimum, and extreme moisture 65% because this is beyond microbial optimum. You become oxygen limited, you start to hinder microbial activity. And so our field site was at the Clarksville, Maryland Central Maryland Research and Education Center. And here I put a map of Maryland with a terribly tiny inset map. And you can actually see the field site here. I threw up some site characteristics in case anyone's interested, but this is on a 3-year rotation of triticale soybean, where triticale is the cover crop, it's a grass, corn grain, and corn silage. We started our experiment right after the soybean harvest in 2024. We had a field day. If anyone remembers last October, it didn't rain a single day. And so we were kind of like biting our thumbs, like waiting, when is it going to rain? So we could get out there and start this thing. Finally, it rained a tiny bit, and we were able to get out there. So we took these aluminum cores that we had made in the PDC. They're 25 centimeters on either side and 20 centimeters deep. We hammered them in with rubber mallets, and we excavated them with shovels, and then hammered in a steel base plate underneath. Put them in a truck and took them back to the greenhouse at UMD. This plot was backfilled with topsoil that the folks at CMREC had. Didn't just leave a hole in the ground. So here's the mesocosm diagram again. I'll discuss the greenhouse setup a little bit. We initially saturated all the mesocosms and let them settle to the desired water-filled pore space. We drilled in seeds to 1.5 inches after 3 days. And we had 2 different cover crop treatments. One was a monocrop of triticale. This is what Clarksville grows, as you saw on the last slide. They use it for feed. Another one was a triticale hairy vetch 50-50 mixture. So triticale is a grass. Sometimes you can use cover crops for a lot of different purposes, but the nice thing about grasses are, in my opinion, my favorite thing about them is that they have deeper roots. They're good for reducing nitrogen leaching. Hairy vetch is a legume. It fixes nitrogen, adds nitrogen to the soil, so that maybe you don't have to fertilize as much in the subsequent crop. So this trade-offs for using different ratios of cover crops, using cover crop mixtures. So we're trying to see some of the differences between doing these 2 practices. We had a randomized complete block design. This is a greenhouse diagram with a really funny, there's actually a PowerPoint background here, this great thing. I think it's hideous, but it makes a lot of sense because this is kind of how the greenhouse bench looked. What do we see here? What is a randomized complete block design? We randomize the treatment's position within each block, and in each block is a replicate. And we shuffled these on the greenhouse bench twice a week to account for greenhouse effects. What's being shown here is the only time you'll see the syntax being used in this presentation, but T and TV, that's triticale or triticale mesh. 35, 45, 65 is the desired water-filled pore space treatment. We water them daily or almost daily, like I didn't work on Christmas, to try and maintain 35, 45, and 65% water-filled pore space. And we measured this via a dielectric probe and using a floor scale. That's what the setup looked like in the greenhouse. And so this is the really cool part of the experiment that I don't think I'm going to talk about enough. So if you want to ask questions about it afterwards, please. So how do we achieve our goal of tracking how carbon is being stabilized in the soil? We use an isotope tracer. An isotope, or I'll talk about it relative to carbon. What's around us right now is 12C CO2, mostly. Okay? And any carbon that you find is going to be 12C. Add one neutron and you have 13C CO2. They weigh a little bit different, and so you can separate them out using mass spectrometry. And that's how you're able to track carbon through different systems. So you're going to track carbon through atmosphere, plant, and soil. So pulse labeling setup, we had our chamber here, threw a collar on it. The reason this collar was on it is because the footprint of these labeling chambers was wider than the mesocosms. I wanted to reduce the amount of volume inside the headspace, because if you have less volume, you can use less isotope to achieve the same effect. Isotope's kind of expensive. There's a little dowel inside the mesocosms, if you noticed from the last slide, that has a little tube going into it. There's a little cup inside that had 5 mils of sodium hydroxide and 13C labeled sodium bicarbonate solid mixture. So you have a basic solution, you add an acid, and what happens? You create volcano. Not really volcanic, but almost like that. Looks like soda. But you effervesce. You cause an effervescence reaction. You release this 13C CO2 into the headspace. You create this labeled headspace that then the plant can take in the 13C CO2, and then we can track it through the plant and soil. What is multiple pulse labeling? It's when you expose the plant to a bunch of short bursts of 13C labeled headspaces. And this is cool because literature shows that it provides a more even labeling of the plant and soil. And labeling soil is pretty hard. If you were to do your own pulse labeling experiment and you labeled one time, maybe you probably wouldn't see any signal in the soil. So doing this over a long period of time, 3 months for us, 18 pulses, would increase the chances of us seeing a signal in the soil, so we can try and cheat out some of these mechanisms. This is what we did. We had the mesocosms on the bench, threw on the collars, threw on the chambers. Inside the chambers, we had an ice pack to prevent fogging, so we can keep photosynthesis going, and a fan inside to circulate the air. Here's Tyler putting in some acid, and then you can watch the fizzy reaction happen. How do we know this is actually working, though? We had a couple of checks. We had a couple of control mesocosms where we hooked up this light work infrared gas analyzer to each time, so in Dr. Baldwin's lab. And we use this to measure CO2 depletion in the control chambers. If we have CO2 depletion happening, we can be pretty confident that we have 13C CO2 depletion happening in the labeled chambers. We wanted to make sure that these plants were taking in the 13C CO2. So here's an example of what we might measure in the light core, and we can assume that about the same thing is happening with the 13C CO2. We also use the Picardo gas analyzer to measure the labeling efficiency throughout the labeling periods. I didn't picture it here, but this gas analyzer gives you pretty high precision ratios of 12 and 13C. And so what I did is I took a syringe at the start of the experiment and a syringe at the end of the experiment every time we pulse labeled. What I was looking for was a decrease in 13C, and pretty much every time we saw it, except when the plants were really young. So now for some sample processing. We had a really fun day in the greenhouse, about 3 months after we first started, where we snipped all the biomass at the root crown, put it in these paper bags to dry in the greenhouse. I had never worked in a greenhouse before this. They're deceptively dry. Who knew? You have so much stuff growing in there. And we excavated the soil to 10 centimeters to get total root biomass for the 10 centimeter zone. We left the edges on because of edge effects happening with this happens with any pot experiment, you're gonna have edge effects. And we put, there's these little holes in the soil here. Those are from when we took moisture measurements at the end of the experiment to try and get what the end moisture was and bulk density after everything was done. So here's this diagram again, and this is why I talked about chapter 1. Also, I think chapter 1 is really cool. But we wanted to figure out what's 13C in these 3 fractions, so we used the fractionation scheme to do it using the optimized energy. We sent these off to UC Davis for isotope ratio mass spectrometry to be done. We got the results about last week. So I'm going to go through some of the results now. There's a lot to dig into here. It's super exciting. Let's talk about what happened with the soil moisture first. All right, so from this, I'll describe what's happening in this plot. You have the average water-filled pore space for the treatment for the entire period on the x-axis. And on the y-axis, you have what's actually the actual water-filled pore space being measured. The x-axis is just like a label, you could say. In the red, you're always going to have triticale. It's not treatment, it's triticale. Sorry, I know it's confusing. And then bet is the mixture in blue. And every plot's going to look about like this. And so we had stratification between soil moisture treatments. These are all significantly different from each other. We didn't quite hit the high moisture target of 65% water-filled pore space, but our stratification will let us know something about what's going on. And it's important to think about this when we interpret the results. Is the above ground biomass in kilograms per hectare? I think megagrams per hectare. I have a better idea of that scale. But still on the x-axis, we have the average water-filled pore space. And then we're going to start to see some of what's happening here. For the middle moisture treatment, around 40%, we have the highest biomass for the triticale monocrops. We have the highest moisture treatment for the triticale vetch mixture. We have the highest biomass. Interesting. I didn't split out triticale versus vetch biomass here. This is just total biomass. But it's interesting to see the differences here. And keep this bar in mind here. Keep this bar in mind here. And this column in mind here, because it's going to be pretty important as we move forward. So now I'm showing you atom percent 13C on the y-axis. This is a metric for measuring 13C. What is atom percent? You can think about if you have a cube in front of you, like the cube idea. You have a cube in front of you, and you have a bunch of carbon in it. 99% of that carbon is going to be 12C. About 1% is going to be 13C. If you have anything above 1% 13C, you have an enriched system. And so we have, this plot shows that all the soil was enriched. This is the total bulk soil for the different treatments. And what do we see? Well, we still see this bar is quite the highest. These are not significantly different from one another, but we do see a little bit of a trend forming where the middle moisture treatments have the highest 13C enrichment overall. But now we're going to split that out between the soil organic matter fractions that we took out. So this is moisture and 13C incorporation into the OPOM. The OPOM, again, included particulate organic matter. You can just think of it as POM. I think maybe I'm putting too many layers on this. But POM is larger material. It's more easily broken down by microbes. And so what has happened here, we see that for the middle moisture treatment for the triticale, we have the highest 13C incorporation into the OPOM. Well, that's interesting because we also have the highest biomass in that treatment. So we have the lowest incorporation of the OPOM for the high moisture treatment for the mixture, but we do see that there might be an outlier down there, right, close to the bottom. So something I want to think about. This is the coarse silt, and I kept the OPOM diagram up here. This is the coarse silt. It's less than 53 microns in size. It behaves differently than clays might, because it's still silts. But we see a trend where it almost appears as if the mixture 13C incorporation is behaving linearly, responding linearly to moisture. 32 to 50% increase in the 13C allocation. But then for the triticale monocrop, we still have the highest 13C in the middle treatment. Also interesting. Okay, same thing with the OPOM, and it had the highest biomass, above ground biomass, I should say. And here's the mound. And the mound is probably the most important fraction. It's the most, it's of interest to us. It's of interest to other researchers because it is a stable fraction. It's what you want to be building in your soil. And we see the exact same trend as we do with the coast-silt, just still plotted down here. Interesting. It kind of makes sense that they follow the same trend. They're both below 53 microns in size. They're both broadly defined as mineral-associated organic matter. So they might behave similarly as a function of their size. These are all three plots together, where we see the y-axis scales are different because there's different labeling efficiencies for each of these three fractions, a function of the weight that you yield from each fraction. You have this linear increase in 13C incorporation for the coastal and mound under the mixture as you increase soil moisture. And then you have, for the high moisture treatment and the lower moisture treatment for the triticale, they're about the same for pretty much all three of these. Then for the middle moisture treatment, you have a spike in 13C incorporation. Curious why. So now I got some conclusions. I guess I talked pretty fast because I budgeted this to be about 10 minutes slower right now. Awesome. A lot of time to discuss. So let's address our hypothesis. This is the same diagram that you saw before. We have our target water-filled pore space treatments, our mesocosms. I didn't discuss these little root diagrams I had thrown in there, but that's just to show the highest mound and pond increase was anticipated for this microbial optimum 45% water-filled pore space. Our trend showed high 13C incorporation and the highest moisture treatment for the mixture. This was about 50% water-filled pore space on average. High relative to the highest moisture cover crop monocrop. Here's the diagram again, where we see this column here for triticale highest moisture is different, pretty different than this column here for the mixture, which I think is really interesting, right? So this tells us something. Maybe this is a function of the cover crop mixture itself, right? Maybe vetch is responding better to higher moisture. Maybe the triticale monocrop is responding poorer for some reason. There's also a lot of other factors we could dig into here, a lot of work that I've started looking at, such as the bulk density. It's important to think about when using undisturbed cores of soil that they are subject to compaction. Let's say, I won't go into it too much. They're subject to compaction, so it's something to think about. Also something to think about is, thinking about the differences between triticale and vetch mixture and triticale itself. It could be a question of root architecture. How, under these conditions, are roots putting organic matter into the soil, so how are they growing? We did not see the highest POM and mound 13C enrichment in the moisture treatments that were around 45% water-filled pore space. That was one of our hypotheses. But we did see that the middle moisture treatment, close to 45% water-filled pore space, usually had more incorporation, she always did, than the low moisture treatment. The low moisture treatment always had the lowest incorporation of atoms at 13C. We did see, I thought it was interesting for this plot, I already told you these aren't significantly different from one another, but we did see in the middle moisture treatment the highest 13C incorporation overall in the total soil organic matter in those middle moisture treatments. It could be telling us something about, it's telling us a lot of things. We consistently saw the lowest enrichment in the low moisture mesocosms. Right, so those 32%. And 13C incorporation, what we're witnessing is it's likely linked to above ground biomass. Literature has told us not too much on this, but it has indicated that more biomass in general, root and shoot biomass, leads to more carbon inputs into the soil. Right? It's pretty easy to think. And so what we have with the highest biomass here, I told you to keep this column in mind, you also have the highest 13C incorporation in the mound fraction. All right, so is above ground biomass linked to mound carbon storage? Above ground biomass likely does not explain all of our results though, there's a lot to dig into here. We just got 13C biomass data for the roots and the shoots yesterday, so I'm excited to dig into that. And hopefully that data will help us link biomass to carbon sequestration and moisture regimes. Trying to link all these factors from our experiment together to create one picture. And we want to ask the questions, we've started to answer them a little bit. To what extent differences in carbon sequestration are a result of moisture? And what proportion of the total newly fixed carbon is going into the mound fractions? Fraction of interest, right? Mound is a hot topic right now. And so with the rest of our data, we might be able to investigate this question of what's going on with the mound in particular more. Hopefully when we see the 13C data between all the fractions, including 13C respired, we can do what's called a mass balance, where we take the fate of all the carbon in the atmosphere, plant, soil system, and we track what went where. We can think about why. Moisture, cover crop regime, bulk density. And so this was my attempt at taking soil respiration measurements, which was like it was fun. I had to stitch these plastic bags together to create this dark room for the chambers to be in. And hopefully we can extend these trends from this mass balance to try and create predictive models for carbon storage rates. And this, our data right now is dependent on our specific methods. But my hope is that these trends can be, I'm not too familiar with modeling, but extended to other systems as well. Hopefully we can contribute to our understanding of carbon partitioning in soil and trade-offs between cover crop regimes. Right? So, for example, the Clarksville folks, they use triticale monocrop right now, and they have a purpose for that. You choose your cover crops with a purpose in mind. The purpose is for feed. But under increased moisture conditions, would a mixture of a grass and a legume be better overall? Increased moisture, maybe you have more nitrogen leaching throughout the winter. If you have no crop growing there, the grass would be good to hold in that nitrate that could be leaching out during the winter. And then maybe you have so much moisture that you're leaching nitrogen too fast. Maybe you just want to build up the nitrogen in your soil, so you add a legume. So I'll mention the first experiment again, and this is a new panel that you haven't seen yet about the analysis that we're doing. We're currently analyzing samples for this project and the other project at the same time. We're using UV-vis spectrophotometry to get qualitative information on how the sonicator is affecting molecular carbon. And this is a big departure from what I was just talking about. I understand that, but I wanted to bring it up. This is the end. And we're also using FTICR, or Fourier transform ion cyclotron resonance mass spectrometry, which is a super high resolution mass spec technique, which can give us information about the molecular composition of all the carbon species in our sample. So we're going to take our extracts from both of these methods. We're going to run them on FTICR. We're going to get plots that kind of look like this, where we can see different ratios. This is called a Van Kervellen diagram, hydrogen carbon to oxygen carbon ratio. We can figure out what kind of biochemical classes are present in our samples. And hopefully we're going to be able to link these to our treatments, our moisture regimes, our cover crop regimes. So references. Thank you to everyone, including every undergrad who's helped me out for 5 minutes or 5 months.
Dr. Ravenhurst.
So this is sort of a methodological question. I'm sort of I think I should understand this. I heard you talk about this a couple of times. Like I got and I got mound, but this coarse silt.
Oh yeah.
So I've always thought of coarse silt as like mineral, but as you're describing it, it sounds like there's carbon associated. So could you explain sort of briefly where this coarse silt fraction fits in a little bit as a carbon containing phase?
Yeah, so our sequential fractionation methodology, I'm going to click back to it. We're kind of going off of some work that was done in 2020 from this author Andrea Gilling. And she split this fraction out because her understanding was it's distinct chemically from the mineral-associated organic matter from the smaller clays. And so it is silts, right? Coarse silt is describing a mineral fraction, but there is also organics associated with it.
So it's when you do the high energy inputs, it's what still doesn't go through the sieve?
Well, we actually do a centrifugation separation. Yeah, we do.
Okay,
yeah.
but it's the coarse
separation.
the stuff that's not further dispersed, high sonication.
Well, we have microaggregates for the high sonication energy. There's probably coarse silt and amount already floating in solution. We also have these microaggregates that we're trying to bust up. And the components, the more dense components of this solution after high sonication is the coarse silt. We analyze that by size. And that wasn't a great explanation. But yes, you have it on the notes.
All right.
Yeah.
All right, so that was the methodological part. And then so I think maybe you addressed this when you talked about you're eventually going to do this mass balance. Because each of these different fractions that you're talking about, I mean, you can get the C13 there, but they also have different proportions of carbon, right? They have different quantities of carbon in there. So the proportion of C13 it's also a function of how much carbon is in there, which you've got a certain enrichment in carbon 13. So that'll come out in your mass balance,
yes.
Right? And put those pieces together.
Yeah. Well, we would normalize to total carbon in the fraction. We have data for all of that. Atom percent 13C is a metric. The formula for that is 13C over 12C plus 13C. So you're already taking the total 12C in that fraction into account. So what's shown here is pretty reliably differences that we can witness between these fractions.
Yeah, so it's clearly enriched on debating
yes.
that. But if there's only a little bit there and it's way enriched, then that's it's different than if you got a lot of carbon there and it's significant. So that'll come out from mass.
Yes, yeah.
So what are you expecting to see from your 13C biomass data, particularly between the different treatments?
So biomass 13C has been analyzed a whole lot more in literature than soil organic matter fraction 13C. Mostly because people want to understand more about plant physiology, so how are crops growing under different experimental treatments. Literature tells us that if you even pulse label once, you'll immediately get carbon down to the shoots. Plants will send carbon down to the shoots. As they take it in, they'll reel it back up if they need it.
You do both above and below ground biomass for the 13C?
Yeah, so we're going to be able to see all these fractions. What do we expect from the different treatments? Well, something related to how much root biomass we have. I don't have the numbers off the top of my head, root and shoot biomass. We can start to think about what causes shoot stress versus what causes root stress and what is the response to that, right? So if we had a really wet system, for instance, roots would be stressed, shoots would probably grow to compensate, okay? And so you might have more 13C held in the shoots. If you have a system that is well aerated, maybe around 50% water for the pore space, you have a nice even labeling perhaps. That's what I'm expecting. But if you have really dense soil, for instance, and roots are very stressed, the above ground biomass is going to respond positively as well, while the below ground responds negatively. Again, root architecture is the thing I mentioned before, and it's not something I've read too much about, but it's an idea that has been floating around, where these different conditions might cause root structures to form differently, and that will also affect where we see 13C in the soil. This is data for the 0 to 10 centimeter section of these mesocosms. Okay, what likely happened is that in the triticale monocrops we had roots that went deeper, and maybe you have more of a even spread of 13C throughout the mesocosm. For these mixtures, maybe you had a lot of the vetch roots. Vetch has like sort of a taproot that goes down, and then most of the roots are laterally on the surface, towards the surface within 5 centimeters. And so maybe you have more 13C enrichment around that area. Yeah.
Great talk, thank you.
Thank you.
I wanted to ask your opinion on another, kind of outlook or use case or selling point for this kind of recommendation, which is pollution mitigation, as you kind of mentioned with the warmer and wetter winters in Maryland, as well as for coastal areas in Maryland where there's ag. Soil is just going to be in contact with water more often. And so it seems to me, and what little I understand, that having a system like this or a methodology for farming practice where there's less artificial nutrient amendment being applied, instead you're using cover crop, maybe there could be some benefit to lowering pollution as well. I was curious if you guys had opinion on that.
Let me know if I do answer your question. Mm-hmm. But in general, like I mentioned, cover crops are chosen for different reasons. One of them could be nitrate leaching reduction. Grasses have deeper roots, they're able to do that. Especially out on the eastern shore where the soils are pretty sandy and you have water going through pretty quick, right? Having a cover crop there to have roots deeper down is nice. But when we say less amendments, it's not necessarily true. Despite having cover crops, farmers will still often choose to fertilize plenty. So, for example, the amount of phosphorus in this soil is pretty huge because they have a lot of phosphorus that they fertilize with every year. And so it would take, these soils are also chisel till, and that encourages the need for fertilization. It would take probably, I think it's land dependent and it's farm dependent, whatever the best practice is going to be. If they need to amend, they're going to amend. And I think cover cropping has the potential to reduce the amount of fertilizer, therefore reduce the amount of nutrients leached into the bay, for instance, or the ocean. But cover crops can do a really good job at supplementing fertilization, but it's not going to account for all of it.
Okay.
Thank you for your question.
Yeah, totally.
I have a couple of methodological questions and related to the roots. How did you separate the roots from the soil from your section that you took it out of?
Yeah.
And can you talk more about the edge effect and what you would have expected in that edge? I assume you did sample it.
Okay, great questions. Thank you. So how we wash the roots is I took this from a couple of papers in the mid-2010s that I figured, like, I was thinking about how would this be easiest to do in the greenhouse. Working with 13C labeled soils, we also do natural abundance work in our lab. I didn't really want to contaminate our lab forever. And so I did it all in the greenhouse. I had a beaker of water. I sieved the soil on 4 millimeters in the greenhouse as I excavated the mesocosm. And then I took the roots and I dipped them in water, swirl them around, aspirate them a couple times on a 53 micron sieve. 50 micron, maybe 120 micron sieve. Aspirate them a couple times to try and get as much of the adhering soil out as possible. And there's still going to be some adhering soil. And especially in the early samples that I took, there was quite a bit of adhering soil. What we did, I don't have a picture here, but we used a methyl furnace to get the total soil, that mineral matter that was adhering to the roots for each of the root samples, and I factored that out of the total measured root carbon, however much soil was still adhering to those roots. And so that ranged from half soil carbon to 1% soil carbon adhering, 5% soil carbon adhering to roots. And then, and a method like this, you're rinsing off a lot of the root exudates. More recent literature, more recent really exciting literature, has shown that root exudates are often unaccounted for in pulse living experiments. Well, I think we did what we could. At least we got root biomass. And as far as the edge effect goes, I have a couple pictures. I took like chunks out of the edges, and you see root matting. Not quite matting, I don't say matting, but you see a lot more root density towards the edges. And this happens because water can flow easier, so it's an easier channel for roots to get into.
So how does that affect your calculation of carbon balance then? You basically don't have those roots in your balance, or?
So we do it on a per area basis. And so whatever the area that we measured, that's the best we could do, right? So we're at the end of the day, we're probably underestimating root carbon a little bit. Something we're going to have to deal with. But we got a good amount of roots at the 0 to 10 centimeters section that we took. And so hopefully, just a little caveat, we can think about that moving forward. Yeah. Yeah.
Quick water question. What was the water-filled pore space when you took your cores? When you said it was really dry, how does it compare to these numbers? And what would, maybe you said this, but what would the number be for like a really wet winter? Is that
Yes.
that high?
So I measured when we first turned in my greenhouse, and they were at about 15% water-filled pore space. It's pretty low. And how would it compare to a really wet winter? So I looked at scan data for the past 25 years. This is Soil Moisture Network from NRCS. And I took an average of the 0 to 20 centimeter section of this project because we have 0 to 20 centimeter mesocosms. Trying to think about like outliers they had. If you get a really wet winter, I'm not quite sure. I can't really give you a good estimate, but I can look into it.
Yeah. Was that is that 65% supposed to be towards that number?
Definitely the upper end.
Yeah.
Right. Soil, 65% water-filled pore space is microbial limiting. Most soils aren't going to hold that much unless there's some sort of restricted layer, something else going on. All right.
So I think we have time for one more question. Spencer, did you?
Yeah, I wanted to ask actually more. Could you explain more about the sonication and the methods of, I guess, breakage that goes on with the aggregates?
Sure.
And that might have implications for things like soil runoff into the bay and what happens to, carbon bound there.
Interesting. I think you asked two questions there. I'm trying to
Kind
answer
of,
both.
mostly on the mechanisms.
All right, so I don't have a picture. There's a great review paper for 2014 that I can send you if you'd like that shows the mechanism in a very simple way. And so ultrasonic waves move through the soil water solution. And Dr. Azursa's lab has also done some work with this, with microbubbles. And so you have aggregates. On the surface of these aggregates, there's oxygen. This oxygen nucleates, or these aggregates nucleate, I shouldn't say oxygen, this air on the surface of the aggregates. Even after they've been put in water, there could be some air, some gas still in there. And these ultrasonic waves will cause pressure differences that will cause these bubbles to expand and contract really quickly. It generates a lot of heat, and eventually when the pressure is too high, these bubbles will burst and release what are called microjets. These microjets can move very fast, they're pretty hot, and they burst apart aggregates by slamming into them. That's the physical part of this method. I mentioned they're really hot. You know what else is hot? The sonicator probe horn. And so this is part of the reason why we're trying to investigate this method for FTICR, or high resolution mass spec. How is this method that we think can do a good job at characterizing organic matter in the soil, is it actually good for that? And then the second part of the question was about field and runoff. Can you explain that again?
Yeah, no, I was just thinking more that the breakage method had to do something more with the physical movement of the particles or collision, which I thought might have implications for stuff moving in waterways, but this is more microjet stuff with the ultrasonic waves than.
Yeah, it's definitely facilitated by the machine
Yeah.
itself. This would not happen in nature.
Yeah, we did, just to kind of follow up on that, we did have this interesting conversation with someone from Pacific Northwest National Lab. When we were first describing the project, they're like, well, have you ever thought about seismic waves? Like from
Earthquakes.
from an earthquake. Could an earthquake potentially dislodge organic matter from soil particles? And I think that's a really cool question. They were really interested in that.
Yeah, there's been some where, sorry.
Yeah, well, yeah, but I don't know if the energy is enough, or maybe it's enough to knock off some. Who knows?
It probably is enough to knock off some, but like you step on the ground and we're knocking off things.
Yeah.
There's a cool 2018 paper about erosion and organic matter depletion after a landslide, and then organic matter accrual following a landslide. In areas like West Virginia where they have landslides all the time, this is sort of relevant because you're constantly shifting soil around. A lot of areas that, what's funny to me is that West Virginia is #1 in erosion because of landslides, and then states that don't really use cover crops much for one reason or another, like Iowa's like #2 in erosion, but you don't get landslides in Iowa. It's just flat, right? It's like their fault. But any physical disturbance like that probably cause something to happen. Right? And an earthquake question is interesting. I wonder how you would even set up a project like that.
I don't know.
Shaker table and a tray of soil or something.
Yeah. But okay, so we should probably stop there because we're at time. Jason, great job.